Good morning, students. Hope you are all safe at home. Today, we are dealing with the balance of introductory part of genetics. The two term you have to study first is wild type and mutant type genes. Then what is wild type gene? A gene when it is found in its natural form. A gene when it is found in its natural form, then it is called wild type gene. That is a gene which is without any change, or we can say that it is a non-mutated form. That means it has not affect any changes. It is seen its natural form. So such a gene which is in natural form without any change are called wild type genes. And these wild type genes are usually designated with a plus symbol. So for any allele we can denote it as with a gene symbol. Along with that there is a plus sign has to be given. On comparing wild type gene, we can say another type gene called mutant type gene. So an individual having a gene that differ from the normal gene or wild gene. So wild gene is a gene which do not have any change. If a gene has undergo some changes due to the effect of some natural or some other effects or some other agencies from outside, or some mutations has happened to a gene, then that gene is known as mutant type genes. So here we study two types of gene. One is wild type gene and another is mutant type gene. Another two terms you have to study is monohybrid and dihybrid. So what is monohybrid? What is monohybrid? Hybrid means hybrid. It is a combination of two organisms. So monohybrid means organism which is either heterozygous or it may be homozygous with respect to only one pair of allele. If we focus on one pair of allele at a locus and saying about only that type of characters, that is character which is determined by one pair of allele at a locus, and if that organism may be either heterozygous or it may be homozygous, then such hybrids are known as monohybrid. Then at the same time, what is dihybrid? It is an organism, we can say that it is either heterozygous or homozygous, but here we considered two pairs of alleles at a locus. In monohybrid, we consider or we take into study about the gene or at a gene which is at a locus there is only one pair. But in the dihybrid, we focus on a gene or focus on the character which is determined by or focus on a gene at a locus, and at a locus there will be two pair of alleles. Such organisms are called dihybrid organism. So these two terms has to study monohybrid and dihybrid. Another two terms, monohybrid ratio and dihybrid ratio. Monohybrid ratio you already studied, dihybrid ratio from lower classes. Monohybrid ratio you have by hearted it as 3:1 and it's the ratio between different kinds of hybrids of the F2 generation, and this ratio we set up from the F2 generations when two parents are crossed and then it will be raised into the F1 generation, and the F1 generation we have raised the next generation, and that generation we called it as F2 generation. And what is the result of that F2 generation, or what is the result we observed in the F2 generation, and that we depicted in the form of a formula or form of a ratio we called as monohybrid ratio. Monohybrid means an organism we considered, previous slide what we say, an organism we considered at a locus, at a locus 1G, or at a locus a pair of alleles are there. Then what is the genotypic ratio of monohybrid cross? Monohybrid cross has phenotypic ratio as 3:1. Phenotypic means, it was already I told in the last class, phenotype means what is the appearance. So appearance, three, or there will be formed four organism, and among the four organism three will be of same phenotype, or three will be of same appearance, and one organism is entirely different, and so the ratio we focused as 3:1 for phenotypic ratio. But in the case of genotype, genotype is the genetic constituent of an organism. Even though the phenotype of three organism is same, when we look into the genotype, we can identify that it is again different. There will be 1, 3, or 3, the number 3 can be again splitted into 1:2. So among the three same phenotype, there will be again difference in genotype. So the genotypic ratio of monohybrid cross is written as 1:2:1. Then coming to dihybrid ratio. Dihybrid ratio is 9:3:3:1. In dihybrid, what we say is there is two pairs of allele at a locus. Two pairs of allele. Since there is two pairs of allele at a locus, in the F2 generation we get, say, how many organisms? 9 plus 3 plus 3 plus 1. So 16 organisms will be we get. So there will be 16 types of organisms. No, we can't say that 16 types. 16 organisms will be formed, and among the 16, 9 will be with of same morphology, another 3 of different morphology, again another 3 of different morphology, and 1 another morphology. So there will be 4 different morphology will be observed for dihybrid ratio. So since 4 different morphology, there is 4 different phenotypes will be observed, and which is written in the ratio as 9:3:3:1. And when we look into details in the number of 9 phenotype, we can again found out that it will be different. And when we look into 3, it will be different. When we look into another 3, it will also different. And so the genotypic ratio is written as, for our easiness, for our remembrance, we can write it as 1:2:1:2:2:2:2:1:1. There will be centrally we can written 4, and in the back and front of the 4, 2. Then same 1, 2, 1. So 1, 2, 1, 2, 4. Again 2, 1, 2, 1. That is the ratio. 1:2:1:2:4:2:1:2:1. Then how many types of genotypes are observed? 9, isn't it? You can count it. 2 plus 1, 3. 3 plus 1, 4. 4 plus 2, 6. Then 10, 12, 13, 15, 1, 16. So here again in the genotype, 16 individuals are there in the genotype, and how many different types of genotypes there will be? 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 types of genotypes are noticed. So in dihybrid ratio, the phenotypic ratio is 9 is to 3 is to 3 is to 1. The genotypic ratio is 1 is to 2 is to 1 is to 2 is to 4 is to 2 is to 1 is to 2 is to 1. And in dihybrid phenotype, there will be 4 phenotype, and in the genotype there will be 9 different genotypes are observed, and among here there will be 16 individuals are noted. Then another term you have to study is hybrid vigor. Hybrid vigor, or we can say the term as heterosis. 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 It is the increase in characteristics such as size, growth rate, fertility, yield of a hybrid. If a hybrid shows much more size, or hybrid show much more growth rate, or a hybrid has much fertility and yield, then what we can say, it is a heterosis, or it is a hybrid vigor, the vigor of that hybrid from the parents. It will be called a hybrid vigor when that hybrid shows much more characteristics than that of parents. Here these hybrid vigor of the hybrids are usually exploited by the plant and animal breeders. How they do that exploitation? They had mated these two different pure lines. Pure lines means when they undergo selfing, the same organisms will be only produced. That means it will not be mixed up. So crossing such two different pure breds, pure lines that having desirable characters, when two best characters of that parents when come together, it will give a vigor to the hybrid. So hybrid vigor, which the term was proposed by, or it was identified by G.H. Shull in 1911. Another term you have to study before going to much more in genetics, that is reciprocal cross. Reciprocal cross. In genetics, we are almost dealing with the cross. Here, the reciprocal cross means the two crosses are there, but there will be a reverse cross. That is, two reversal cross in which sex of the parents are interchanged. How the sex of the parents are interchanged? In the case of plants, if the pollen from tall plant is transferred to the stigma of dwarf plant in one cross, in the reciprocal cross, what will be considered? The pollen of the dwarf plant will be considered. So, once again, reciprocal cross means reversal of the reverse cross with the sex of the parents. For example, if the pollen, that means that plant is considered as the male. If the pollen from tall plant is transferred to the stigma of dwarf plant in one cross, and in the another cross, or in the reciprocal cross, the pollen of dwarf plant is considered and the pollinated to the stigma of tall plants. Here the picture is depicted. You can look into that for more details. Then another term is back cross. What is a back cross? Back cross means mating of a hybrid organism with one of its parent, or with an organism genetically similar to the parent. It is the back cross. crossing back with the parents or crossing back with an organism genetically similar to the parent. That is the back cross. The back cross here is the in pictorial form it will be depicted, that is capital Y which gives an yellow colored seed is crossed with the small y, that is with the green colored seed we get capital Y because capital Y is yellow colored, and here the capital Y, presence of capital Y gives the F1 organism as yellow colored seed. When we cross the capital Y small y with another or with an individual same as that of the parent, which is small y, what we get capital Y small y in the ratio 1:1. So the F1 progeny is back crossed with the one of the parent. It is either of the parent. It is called back cross. Then about test cross. Test cross are used to find an individual's genotype. Test cross is for identifying the genotype of an organism. How that genotype of that organism is identified? It is done by crossing that unknown genotype organism with a known genotype. Here in the back rows we get in the F1 an yellow seeded fruit. We can't know that if that yellow seeded fruit is with the genotype capital Y or capital Y small y. In order to find the genotype which is correct, either capital Y or capital Y small y for the yellow seeded fruit, we have to cross that F1 with another genotype, another individual in which the genotype is known. Genotype is known by us. So, test cross reveals whether an individual is homozygous dominant or heterozygous. So, the test cross helps to find out if the F1 is with the homozygous dominant, that is with capital Y or with this capital Y small y. I am comparing this with the examples from the back rows. Again, monohybrid test cross ratio is 1:1, but dihybrid test cross ratio is 1:1:1:1. That means dihybrid means there will be two genes which assorts independently. If one genes are there, then the ratio is 1:1. If two genes which assort independently, then the ratio is 1:1:1:1:1. 4 phenotypes can be observed. Here example for test cross ratio or test cross, that is capital D into small d. Here the individual is capital D small d. We can cross that capital D small d into small d because here the homozygous dwarf small d, we are sure that the dwarf has the homozygous condition and the dwarf has a recessive condition. So we are crossing the unknown genotype tall plant with a non-genotype dwarf plant to identify whether that organism is capital D small d or capital D. Since in the test cross we get one tall and one dwarf, we can show that in the F1 there is a homozygous or there is a recessive gene, because an allele from one parent and another allele from another parent has to come in the hybrid. Since there is one small d in the F1, then only we get an offspring which is dwarf in the F2. So here when the heterozygous or when the F1 is crossed with the homozygous dwarf, if the F1 is with a homozygous dominant, then we never get a dwarf individual in the F2. But here we get one tall and again one dwarf. So since a dwarf individual we obtain, we can show that there will be a small or there is a recessive allele in the F1 tall plant. That is the way test gross helps to identify the genotype of the unknown organism. Then what is F1, F2, F3 generations? And the term was coined by Bateson and Saunders in 1902. F1 is the first filial generation, that is generation of hybrid produced by crossing between two parental generations. If the two parents are crossed, we get the first generation, that we call it as F1. It is the first filial generation. If it is raised to second, then will be the second filial generation. If it is raised to third generation, we call it as F3, which is called third filial generation. Then another term is Punnett square or square checkerboard. When we write these genotype and a phenotype in a ratio method, if the number of genes in a locus is, a number of alleles in a locus is more, we already know that in dihybrid there will be 16 different individuals. To write it in a linear way, it is very difficult to identify what are the same type of genotype, what are the different types of genotype. Then it is in the 20th century, that is much after Mendel's experiments on genetics, this tool was created. And that tool is called Punnett square. It is a chart that helps to determine the expected percentage of different genotypes in the offsprings of two parents. It is the expected genotype or expected percentage of genotypes in the offspring. The gamete produced by the male parents, and how that Punnett square is written. The gamete produced by the male parents are at the top of the chart. Gamete produced by the female parents are to be written on the side. The different possible combinations of alleles in the offsprings are determined by filling the cells of the Punnett square with the correct letters. Here the letter means allele. You can go through Punnett square table of monohybrid cross. That is pollen, and the pollen, the two gametes are capital B, small b on the top of this table, and from the female plant pistil, from there are two gametes which is written as capital B and small b on the side, and in the each square we can fill it with the letters or with the alleles, that is above and below, capital B, then capital B, small b on the top right, and small b, capital B, small b in the bottom left, and small b in the bottom right. If there is four alleles, two pairs of alleles, we can extend this column, and in the monohybrid there will be four different individuals will be formed, four individuals will be formed at maximum, and in dihybrid cross there will be 16 individuals will be formed in maximum. So Punnett square table for monohybrid has with 4 squares for filling the hybrids, but in the case of dihybrid cross it will be of 16 squares for filling the hybrids. So these are the different terms you have to study before dealing with the genetics. So students, today we deal with so many terms. Most of the terms are familiar with our students, but you have to understand in a thorough manner what these terms deals with. Then only we can go into much more details on genetics. From tomorrow on, or then on the next class onwards, we can start the classical genetics, or classical genetics means Mendelian genetics, or Mendelian genetics. Now I am stopping here. You all prepare notes and study well. Thank you.